All right. I think did we not start a video already? Okay, so we're going to continue from our last video where we introduced the idea of the codons and the different parts of the, I think we ended up with the 3 kinds of RNA that we talked about, right? Okay, so now we're going to do sort of stage 1, which we're going to call transcription. And this is the process of getting the information. We're literally going to transcribe, which means to sort of copy over, right? When you transcribe something, you copy it over. The Bible has been transcribed many times in different languages, and books are transcribed. The courtroom events are transcribed, turned from vocal interactions into written interactions by the court reporter, and so on. So essentially what we're going to do in transcription is we're going to copy the DNA information onto messenger RNA, which from now on I'm just going to use this shortcut, mRNA. So you have to remember, right, that's messenger RNA. Onto messenger RNA for transport to a ribosome. That's the first step. And then what happens at the ribosome is the next step. Okay, so I guess what we'll do is we'll draw a picture as usual. Oh, did you need some more time? I'm going to use a series of different pictures. Your textbook has some okay pictures, but some of them are a little bit odd. All right. So we're going to draw a section of DNA. I'm going to draw. Well, I guess I'll draw the DNA in black. Well, maybe not. Maybe we'll do the DNA in blue. How's that? So the DNA strand is going to run something like this. And it's going to be sort of, instead of being split entirely open, it's going to be sort of bubbled out like this. And then we'll draw it along like that. This will be the 5' end, and this will be the 3' end of the top section of DNA. And then the bottom section of DNA will be coming along, and it'll be sort of popped open as well. And this would be running in the opposite direction because it's just a regular DNA piece, right? And essentially what's happening here is this large sort of enzyme complex. We actually call it a transcription complex. It's many enzymes working together. At the start of this section that needs to be transcribed, there are codons, or there are sort of bases in sequence that indicate where to start. And so we call this section of the gene the promoter. So I'm just going to put in red here, and let's see, which one are we going to copy? We're copying from the 3 to 5 direction. So yeah, we're going to copy. The bottom piece is the section that's going to be copied, because remember, the enzymes only move in one direction, right? The enzymes ride along in the 3 to 5 direction. They build in the 5 to 3 direction. So the enzymes can only go towards the right-hand side, from left to right. So they have to ride along the bottom strand, and they can't really ride along the top strand as easily. So when the complex separates this, it has to move in the direction of the bottom strand. And so we get right here, I'm going to put, well, this isn't in the picture, but I'm going to just put a little sequence here. This little sequence here is called the promoter. And the promoter, well, it promotes the process, or starts the process. And the promoter contains a sequence of bases, in particular, thymines and adenines. And so in eukaryotic cells. It's different in, we have to understand that the biochemistry of bacteria and prokaryotes is slightly different because it's far more primitive, right? So adaptations have been made by evolution as the development of eukaryotic cells came about. So in eukaryotic cells, there's something called, we call it a Tata Box. And all that is a sequence of T's, thymine bases, and adenine bases. It might be, you know, TTA, TA, TTA, TTT, or something like that. Just a bunch of them. And the thymine and adenine, if you go back to your pictures on how the bases actually bind with each other, with those hydrogen bonds, you'll find that the thymine and adenine only have two locations where they hydrogen bond, whereas the other ones have three. So this is an easier way to, or an easier spot to break open the DNA. So if you have a section of DNA that has a whole lot of T's and A's, it will be a relatively easy spot to pop open. So that's why it becomes the promoter or the beginning part of the sequence. So the Tata Box is the sequence of thymines and adenines that is easy for the enzymes to pop the DNA open. So it serves as a way to indicate the beginning or the start. So the Tata Box is actually just, it's right on there. I'm just, the red box isn't a thing, it's just highlighting that section. So you'd have, basically, along that blue DNA, it would be TTA, ATA, TTA, TTTT, or something. There'd be a large chunk of the DNA that was just A's and T's. And that's what the Tata Box stands for, okay? A high concentration of those, which means that spot is easy to open, and so that's where it begins. Okay, so there are proteins called transcription factors. And these transcription factors will recognize the promoter and they will open up the DNA and sort of get everything going, okay? All right. So let's just do that. Let's put a little bubble here. Let me be in purple. So let's just put a circle here, a purple bubble. And this purple bubble, I'll color it with a crayon so it's kind of shaded. Lands on the promoter. And it's essentially going to allow the RNA polymerase this time, which is going to build the RNA strand, to bind. And there could be more than one of these transcription sort of, these guys are helpers, right? They're sort of, and but what I mean by that is if you just think about the whole biochemistry, in order for things to bind and attach, there has to be changes in shape, right? So that active sites on these molecules can be exposed. And so each of these is sort of doing its thing to make this happen. So let's just put a little line there. We'll call that a transcription factor. So that's the transcription factor. And there could be, I'm going to put an S on this because there could be more than one. There could be several that are binding in this area right around the beginning to make this happen. All right. The transcription factors allow the binding of the big RNA polymerase II. So we'll put that in maybe greenish here. And the RNA polymerase II is a very large molecule. And so it basically engulfs all of this. Like this. It engulfs the whole thing. So let me crayon that in. Okay, there's the RNA polymerase II. So I'll put that up top here. RNA polymerase. So you remember DNA polymerase built DNA strands on the open section. RNA polymerase is going to build RNA strands, and it is two, or they usually use Roman numerals. Maybe I should do the same. polymerase 2. Doesn't really matter. You can use a numeric 2 if you want. And so the polymerase expands, helps to open up the DNA, and what it begins to do is it begins to assemble at the front end here a piece of RNA. So now we need another color. Perhaps we'll use, what do we got? Let's go back to our funny little orange color. Let's try that. So right along here, there would be the beginnings of, a little thicker, the beginnings of an RNA molecule being built. So let's just put RNA over top of this. And of course, this is going to be the messenger RNA. It's being assembled. Now, as this thing moves along, it starts to create this RNA chain. But eventually, the RNA reaches the end of the sort of bubble or the complex. And then what happens is, of course, my picture has words all in the place where I'd like to draw. But what happens is this just gets strung out like this. And it starts to hang out of the complex like a big long thread, because the complex is also closing the DNA behind it as it goes. And so this just can't stay attached, right? So this big long thread of RNA is being slowly made as this entire process sort of slides along, all moving sort of in this direction along the DNA. And it's assembling RNA. So we could put, like we did before, the little base, the little lines to show the bases in the DNA molecule. Remember how the ladder structure like this. So these would be open. And then on the bottom, it's building RNA on top there. And remember, the difference with RNA is that it has uracil and it has ribose sugar, and it usually forms a single strand like this instead of doubling up with another. And that's essentially the process. So the RNA polymerase moves down. The promoter contains the Tata Box, which is the starting point. There's also a termination spot. When you get to a termination codon that says stop, remember how there are start and stop codons as well? Then this RNA polymerase will just detach and the whole thing will stop, and then the messenger RNA will just end at that point. So it uses the codons to determine when to start and when to stop. But the starter promoter also has not only just the start codon, but also the Tata Box, which is a helpful way to get the RNA polymerase to attach. And the transcription factors are smaller protein complexes that can locate those things and give the RNA polymerase more of a grip on the whole thing, so that's where it knows to begin. So that's kind of the picture. That's the process. Now, if we look at the piece of RNA that we've just copied, right? If we look at it in detail, what we'll find is this. So I'll use that same color. So this piece of RNA, thicker line. This piece of RNA that's made, sometimes we call it pre-messenger RNA because it's not really ready to be the messenger yet. It contains the bases and things. And we'll stick those on one side here to show the A's, G's, C's, and U's now. Remember, the U's will replace wherever there was, where there would have been a T. And if we look at this thing, we'll find that it has at the beginning, right? Well, let me just get a better picture here. Okay, it has sections. So I'm going to use a blue here. This section here will be called the exon, and there's be an exon over here. Exon. exon. And then the other parts, this section is what we call an intron. Intron. The introns and the exons are essentially the difference between the important coded parts that we need to make the protein, those are the blue exons, and the introns that are in between are these other segment sequences that do not code for protein. They're just kind of in the way. They're almost extra little bits of gibberish. And as I said, they probably evolved as a way to protect or at least enhance or lower the chances, I guess I should say, of mutations actually affecting the important parts of the code. So we inserted Mother Nature inserted all these genetic sequences, or these, I guess you could call them not genetic, base sequences, because they don't code for genes. We really don't know what their purpose was when we first discovered them. But now we theorize that having all these little extra bits means that if a mutation happens, there's a higher chance it'll happen to something that we don't need or care about, right? So there's these extra pieces. And so now there's some processing that has to happen to this molecule, some processing. It has to be snipped apart. The intron, well, should we write that down underneath, just so you don't forget? Introns are the extra base sequences that are not part of the gene code, or not part of the gene code, let's put it like that. And then the exons, these are the important, well, not important. I shouldn't say that they're any more important. The other ones have an important job. It's just not they're not part of the genes. So let's just say it's the part of the code that codes for genes or for proteins, for specific polypeptide sequences. So in other words, the blue exons are what makes up the genes, and the introns are sort of the extra bits in between. Okay, so first we have to splice and dice here. We have to cut this molecule apart. Everywhere there's an intron, snip it out, and then rejoin it together. And there's a whole other process that takes care of this. There are these things that are called, we call them Snurps, and they're called small nuclear ribonucleoproteins. So let me just write here small nuclear, which means they're in the nucleus, ribonuclear proteins. So they're complexes made up of RNA. Ribonuclear refers to like the ribonucleic acid part, and proteins that are stuck there with them. Yes? Yeah. Go up. How's that? Okay. The small nuclear ribonuclear proteins. So the shortcut for, sorry, it keeps doing that. The shortcut for that is little SN, small nuclear ribonuclear protein. And so they've been affectionately called Snurps, instead of using that big acronym all the time. S-N-U-R-P-S, Snurps. And these are smaller complexes that will actually bind and locate the introns, snip them out, and then rebind the blue pieces. And so if I drew a picture of that, what would happen is several Snurps would get together, and they would form a complex again. So let's draw our DNA. What I'm going to do to draw it is draw it sort of, what color were our exons? Blue? Yeah. So we'll draw an exon piece here. And what happens is the spliceosome sort of loops this around. This is the intron part. And then the exon part is sort of carries on like this. And so what we have is this large sort of structure here. Crayon, purple. Okay, I thought I pressed purple. Obviously I didn't. Purple. I'm just going to lightly shade that in, purple. And inside this complex there will be several of these Snurps. And the Snurps contain RNA that's all twisted up, bound by proteins. So we'll draw in a couple of these Snurps in another color here, maybe green. So there would be a sort of a Snurp. We'll put 4 of them in here like this. 1, 2, 3, 4. And if I shade those in green, those are the Snurps. So you might want to do some labels. Yeah, so the green, let's label the green Snurp. And the purple, the whole thing is called the Spliceosome. The word zome, the suffix zome, basically just means a body or like a chunk of something, a lysosome, a ribosome. It means a blob, essentially. And so this spliceosome is the blob that's going to be cutting and splicing the DNA. And so essentially what it does is it recognizes this intron, it snips it out, and then it cuts it and puts it back together. That's where you can see the folding. The reason it's folded is because you can imagine these two ends are being brought together, and then eventually we end up with a nice piece of spliced DNA, or RNA, sorry, without the intron involved. So that's the first thing that happens. The next thing that happens, well, I'm not sure if that happens first or second. It doesn't really matter which happens first or second. They kind of both probably happen somehow at the same time. But the next thing that happens is that the ends of the DNA are vulnerable. So the next thing we'll write is the ends of the messenger RNA are vulnerable. What that means is vulnerable to reactions with other molecules. They're fairly active. And if they're active, that means that as this, all right, so everyone wants a label on the exons here. And in very small red, doesn't listen to me, I'll put the intron down here. There you go. So we're snipping out the red piece, right? Over here I use the blue and the red. Now you got it? Okay. And so got the picture? And we're down here and we're talking about the next thing that happens, or another thing that has to happen, is that the ends of the DNA are highly vulnerable to being reacted with. They're fresh, right? They've been cut out. And they're just kind of dangling. The ends of the RNA, I should say. That's what I meant to say. And so we have to do something to them to sort of seal them up so that other molecules won't react with them. Or if they do, they won't damage any of the important message. So there's two things that happen. We end up with our strand here of just exons have been snipped. And then in another color, maybe I could use this cyan. No, it's too close. Maybe this bright green color will be nice. So what happens is on this end, there's a little addition or a little section Now I should have labeled this, so let's go back and label the ends. This is the 5' end of the blue molecule. This is the 3' end of the RNA. So what happens is this little section here, this little blob or thing gets attached onto this end. And it's called the 5' cap. That's what it's called. And it's just a sequence of bases that sort of sticks on there and sort of makes that end less vulnerable to attack. On the other side, we get a whole sequence attached over here. Although it's different, because the 3' end, those both look like 3's, don't they? I just noticed that too. So let's clarify that. This is a 5' end, and 3' end. So the 3' end is chemically a little bit different than the 5' end. It has different sort of parts of that ribose sugar that are hanging out there, so it has different bonds. So this end is called the poly A tail. So let's label here. Let's label the 5' cap, 5' cap. That's this piece. And over here, we get the poly A tail. And what it basically is it's a whole sequence of adenines. It's a whole bunch of A's, adenines that are added on. So if they get attacked, okay, if you have 20 adenines stuck on the end and a molecule comes and reacts, well, it'll just take a few adenines away, and it's not going to interfere with the precious blue exons, the gene itself. So the poly A tail is another piece of modification that happens to the messenger RNA, and this is to protect it. So these are the two important steps that have to happen after we go all the way back here to where it's copied by this complex. The RNA is created, but it contains both the introns and exons, and therefore it has to be sort of edited. Introns have to be snipped out, and it has to be spliced together, and we also have to add these two little caps. And now the RNA molecule is all grown up, properly trained, and ready to leave the nucleus. So now we are ready. The final messenger RNA is ready to move from the nucleus through the nuclear pores in the nuclear membrane. Nuclear pores. And remember, these nuclear pores are necessary because these are big molecules, right? RNA, they're big, long, strandy molecules. It's very difficult to transport these the way we've learned through protein gates or through, even diffusion through a membrane. It wouldn't work. So the nuclear membrane has to have larger holes called the nuclear pores where these molecules can get through. And when they come through the nuclear pores, they end up going into the endoplasmic reticulum. Endoplasmic reticulum. That spelling is, I'm going to blame it on my writing. That says endoplasmic reticulum. We can put ER for short if we want, now that we're all cell biologists after grade 10, right? So endoplasmic reticulum, and then in the endoplasmic reticulum, we find near the nucleus, the rough ER, we find the ribosomes. So now that's basically where everything takes place after that. The protected, copied piece of messenger RNA goes off to the ribosome, leaves the nucleus behind. And we'll stop this video here, and we'll then do a second video with the next part, the translation part, where we actually build a protein.